So, hello everyone. Good evening. Welcome to week 5 of problem solving session. So, before I begin, I would like to know if you are guys are done with week 4 assignment. Also, have you started attempting week 5 assignment? Hello? Am I audible now? Hello? I disconnected for a few seconds. Am I audible now? Can you guys hear me? Is my screen visible? Hello? Kavya, Vandana? If you guys are in the meeting, please respond. I'll assume that you guys are able to hear me. If you face any issue in hearing or something like that, please let me know. My screen is visible, right? So I hope that you have successfully completed your week 4 assignment. Okay. If you face any doubt in solving those problems and all, let me know. So in today's session, we will discuss a few problems related to week 5 assignment and related to week 5 content. Okay, so a brief introduction about myself. I am Chandra. If you don't know already, I have been taking this problem solving session for past 4 weeks. This is the fifth week of the course. And I am a PhD student at the Department of Chemical Engineering at IIT Kanpur. And for my thesis, I am working in the area of tissue engineering. Like every Saturday, we will meet till I guess 21st September or week before that. I'll try to take one revision session as well before your exams if you have enrolled for the exams. And all the notes are available in a Google Drive, and also the videos can be accessed in my YouTube channel. The links are given to you already. So for this meeting, you won't be able to access the chat option. So if you have any queries, I request you to please unmute yourself and ask directly. And interrupt me anytime, whichever point you are not able to understand, interrupt and ask. Okay. And you can also use the hand raise option for interaction. So for the past 4 weeks, we have been extensively studying about biomaterials, right? And what are the properties of biomaterials? What kind of biomaterials were there? How to characterize those properties? Now, this week onwards, we are going to look at the next aspect of tissue engineering, that is cells. So, given in this slide is the tissue engineering triad. So, biomaterials, cells, and signals. If we are able to achieve a balance between these three components, we would be able to create an artificial tissue whose properties are more or less similar to the native tissue. That's the whole idea of this field, right? So, when we're talking about cells, we have to first consider what type of cell we need. So that will basically depend on what type of tissue you're targeting. If you are targeting skin cells, so skin tissue, so you would want cells like keratinocytes, fibroblasts. If you are targeting, say, heart, heart tissue, then you would want cells like cardiomyocytes, etc. So, depending on what tissue or organ you're targeting, you would have to choose a unique cell type. Okay. Also, one organ or one tissue does not necessarily have only one type of cell. So, it's a multicellular organ or a tissue you know already. So, to mimic the actual function of a native tissue, we have to introduce multiple cells at a time. Okay. So, depending on the tissue type, you will choose the cell types required. Then we have to think from where will we get these cells? So, what are the cell sources we have to ponder upon? So, the cells can be isolated from your own body, that is autologous, or you can take it from a member of your species, the allogenic. You can take it from an animal, xenogenic, or you can take it from a identical twin, who is genetically identical to you. So that is syngenic. So depending on what source you're taking, you can classify accordingly. Then also, since suppose you identified the source, then you have to consider what type of cell you need, whether you need differentiated cells for your application, or you would need a more advanced type of cell, that is stem cells or reduced pluripotent cells. Okay. So depending on what your target is, you would have to choose the cell type and cell source. Then you have to think on how to harvest it. So how would you get these cells from your body? And in single suspension form, ultimately you want to get specific cells, and each of the cells is identical to each other. Means you want a homogeneous population of cells with identical properties. Ultimate goal is that. So that's the starting product, homogeneous population of the cell for seeding it on a biomaterial scaffold. So you have to think of the harvesting process that will help you achieve that technique. Then once you harvest, say, a crude cell population where there are multiple cells types present, okay, of different sizes, shapes, properties, then you have to go for specific techniques that will allow you to isolate specific cells. Okay, so that is cell isolation techniques we will look into. Then, when you're familiar with all these things, you would then have to understand how to uh, mimic the function of the tissue in vitro, means on your artificial tissue. So, in order to do that, you have to understand how the tissue works. Okay, tissue is not static in nature; it is very dynamic. Means every time few cells are dying, a few cells are undergoing differentiation. Okay, so there is a dynamic state exists in that tissue. So how to mimic that? Okay, so in order to learn how to do that, we have to first understand what is tissue dynamics. Okay, which properties of cell behavior ultimately contribute to the dynamic property of a tissue. Okay, so for today's discussion, I'll stick up to cell isolation technique. Okay, from next week onwards, week 6, I'll go more in detail about tissue dynamics and under which how many cellular, so there are under tissue dynamics, there are various types of cellular fate processes involved, like as I said, cell death, okay, cell division, cell proliferation. These are all cellular fate processes. Like at any particular point in time, within a tissue, the cells will undergo either of the different types of cellular fate processes, like cell division, cell differentiation, cell death, cell apoptosis, cell adhesion, cell migration. So all these things are happening, okay, at any particular point of time in a tissue. So that's why a tissue is in a dynamic state. So I'll discuss more on tissue dynamics and cellular fate processes from next week. Okay, during next week. Today I'll only discuss problems up to cell isolation part. Okay, whatever professor have discussed up to cell isolation. So now Before I begin the problems, so I have large amount of at least I think good number of problems I have with me today. But before that, if you have any query regarding anything that I have discussed so far, any doubt? All right, I'll move on with the first problem. Which of the following, oh, so this is the first problem. Which of the following is or limitations of xenogenic or and allogenic transplants? So the question is asking the limitations of xenogenic and allogenic transplant. Okay. So there are multiple options can be correct here. First option is disease transmission, then immune rejection, limited availability, limited immune rejection, limited availability, donor side morbidity. So either one option or more than one option is correct here. Which of the option is a limitation for xenogenic and allogenic transplants? So xenogenic transplant refers to transplant derived from animals like porcine, pig, bovine. Okay, from these animals, many a times people use their um, say bladder section, urinary bladder section to suture into a body or decellularize it, okay, and use the ECM porcine, decellularized ECM of from porcine skin or bovine, and then use. So these are all xenogenic. Allogenic transplant means it is derived from a same species, like we are all humans, right? So it is not taken from my body, but maybe it is taken from a friend or my a husband or my father or my mother. Okay, so same species the transplant is derived from. Okay, so what could be the limitation when you are using this kind of transplant? So xenogenic or allogenic transplant means it is not belonging to your own body, as the name suggests. Okay, so what it can cause, it can risk the uh, transmission of some foreign disease, maybe from another person or from another species, right? So this option A could be a limitation. Also, whatever components are there in those transplants, if it is not decellularized, it is cellularized, then it can uh, trigger immune rejection because these transplants will contain foreign antigens, which our body's immune system can recognize as foreign, okay, and elicit strong immune reaction against it. So immune rejection can happen with these transplants. whereas availability and donor side morbidity is not the primary concerns when you're talking about xenogenic and allogenic transplant. Okay, any questions? Okay, moving on. This is the next question. Which of the following is a concern for which of the following donor side morbidity is a concern? Okay, so there are some situations from like option A, B, C. You are isolating cells, okay, from different sources. So in which of the option donor side morbidity is a primary concern? Anyone can tell me what do they understand by donor side morbidity? What this term mean to you?
Ma'am, am I audible?
Yes, yes. Am I audible to you?
Yes, ma'am. Ma'am, in order
Okay.
to fix the repaired or the affected tissue, we scrap or extract the tissues from the region where it has nothing to do with the affected region. So we take the tissues from a clean region and then we place it here. All the same, we also have an affected area in the place where we scrapped the tissue.
Yes. You're in order to solve one problem, you create another, basically here.
Yes.
Right? So that is donor side morbidity. So in which of the options would you cause donor side morbidity? Option A is allogen. You are trying to isolate allogenic cells from a cadaver, then autologous cells from the patient, xenogenic cells from animal tissue, syngenic cells from identical twin. Also, note the term donor. Okay, who is the donor here in this case? The same person from who is having the injury. Okay, this term is usually used for those cases. Anyone? So when you are isolating allogenic cells from a cadaver, say human, obviously human cadaver, so that body is dead, right? Anyway, it's going to be incinerated or whatever, buried or something. And with proper clearance, you are isolating some tissue or organ piece or something like that in order to get cells, right? In autologous cells from the patient, what you are doing, you are maybe taking some bone marrow aspirate from that person, from his own bone marrow only, so that you can use those cells from bone marrow to you later on use to transplant on that patient later, right? Maybe seed it on a scaffold for creating an artificial tissue, and you will later on use that artificial tissue uh, on that patient. Okay. Here, xenogenic cells means porcine cells, bovine cells, rat cells, something like that you're taking. Syngenic means the person who is the patient. has fortunately an identical twin. So that person is genetically similar to that person. So you can take the cells from that twin and then use on the patient. So which of the scenarios, for which of the scenarios donor side morbidity is a concern then?
Autologous.
Yes, you're correct. Yeah. So in case of say patients who is having burns, third-degree burns and all, doctors usually scrap, take a small tissue section, skin tissue section from the thigh region, okay, and transplant it on the burnt area. So while doing that, they also create a big wound on the thigh. So that's a case of donor side morbidity. This is the next question. Stem cells are differentiated and divide in a definite manner and give rise to specialized cell types. As stem cells undergo symmetric cell division. Is this statement true or false? So, you guys know what are stem cells? Stem cells, if I were to define in very general terms, these are the cells with high potency, means very high ability to divide. They can undergo theoretically a large number of cell division. Okay, they are what do you say? Um, can differentiate along different lineages. So they can differentiate along different lineages and give rise to different types of differentiated cells. Um, Also, these cells can self-renew, means um, they can maintain their population. Okay. Um, few cells will undergo differentiation and replenish, say, differentiated cell population, whereas in the process, few cells stay back in within the stem cell niche and maintain the population of stem cells. Okay, and these are like found in various difficult or very unique, discrete regions in different tissues in our bodies. People are still researching to find these niches and isolate the stem cells. So the number is very less in relating to other cells which are there in that particular tissue. Okay, so they're found in very less number, but these cells are there in your tissue throughout your life in order to help you repair and regenerate any defect in your particular tissue or organ. Okay. So there are different kind of stem cells also. Depending on potency, there can be multipotent, totipotent, pluripotent. Okay. Um, as I said, they can undergo very large number of cell division. Also, differentiation does not happen in one step. Differentiation uh, at the end stage is mature cells or differentiated cells. So from stem cell to differentiated cell, it takes place through several stages. Okay, so there can be stages like early progenitor cell, late progenitor cells, precursors, and like that. Okay, so people name it differently. And each stage of cell type has a different characteristic. Okay, so in mota-moti, this is what stem cells are. Now, this is the statement in give that is given to you. Stem cells, they can differentiate and divide in a definite manner. Okay, note this. Is this correct? Is this correct? Can they divide and differentiate in a definite manner? and can give rise to specialized cell types. So this is right, right? This statement is right. This part is right. Stem cells can differentiate and give rise to specialized cells with specialized function. And stem cells can undergo symmetric cell division. Now, is this part correct? This part and this part. If you are able to guess whether these two parts are correct or not, then you can answer whether this statement is true or false. So first question here I'm asking, can stem cells undergo cell division in a defined manner? Or can or is the cell division uh, can happen uncontrolled or over a large period of time, over a large cycles? First, next question, does the cell division that happens, does it happen symmetrically or asymmetrically? So symmetrically means um, the daughter cells that are formed, they are identical to each other, just like in mitosis or meiosis. Whereas asymmetric means the daughter cell differs from each other. So one daughter cell will have a different property, another daughter cell will have different property. That is asymmetric. So the question is, does the stem cells undergo symmetric cell division or asymmetric cell division? Give it a thought and let me know what you think about it. Then I'll give you the answer.
Asymmetric cell division.
Yes, the stem cells will undergo asymmetric cell division. Why? Because the cells want to restore or maintain its population. Okay. Here, what happens in asymmetric cell division briefly? This is a parent cell. Okay. And two daughter cells are formed. Now, it's possible either these two daughter cells is case one, case two, case three. Now, in case one, what will happen? These two daughter cells are differentiated. Then, if both are differentiate, then what will happen? There will be loss of one stem cell from the entire population, right? So this will be termed as symmetric. Case one will be termed as symmetric. Now, case two, what will happen? One daughter cell retains the stem characteristics or stemness, it is called, whereas other becomes differentiated. So then what happens is that the daughter cell, which is having the stemness, will now replenish the stem cell population. Okay, this is asymmetric. And third case, what can happen? Both the daughter cells are stem. All right. So this case three actually increases the cell population. So usually case two is observed. All right. So that is why cell division in stem cells is asymmetric in nature. And oftentimes the cell division, what happens? So before the daughter cell, one of the daughter cell undergoes differentiation, it will first proliferate. So there is a stage of proliferation first. First, the remember always from stem cells, always proliferation will happen first. Okay, that will generate a large number of cells, and then each of these proliferated cells will undergo differentiation, which is why even if the tissue is having very small number of stem cells, it can help giving back a large number of differentiated cells. Okay, because differentiated cells themselves do not have that much cell division capacity. Okay, so it's possible that the cell cycles, number of cell cycles is not defined. It will be very large. So depending on the age, depending on your age, uh, the cells division number can increase like 50 to 30 doublings. And as you grow old and old, the cell cycles that the stem cells can undergo will decrease like 10 cycles, 20 cycles. Okay, so it is not definite or it is not fixed. The division, cell division is not fixed. Okay, so that is why this is also incorrect. So this statement is then false. All right, is it clear? Is this question clear? I'll assume, okay, that you have understood. If you have any queries, let me know. Okay, I'll move on. Okay, this is the next question. Why is the use of stem cells becoming more popular in tissue engineering? Select all that apply. Okay, so there is more than one option that is correct here. So in tissue engineering, why are people more and more becoming interested in using stem cells? So this is just help you understand the importance of stem cells in this field. Okay, the first options is differentiated cells have limited cell division capabilities. That's true. Okay, then differentiated cells can be easily harvested in large numbers. Is it correct? Is this statement correct? New sources of stem cells are being identified. Yes. And stem cells can give rise to a large number of differentiated cells. Yes. Okay. So now tell me, is the option B correct? What do you think? Can we harvest differentiated cells in large numbers? Like large number means in the order of 10 to the power 6 to 10 to the power 8. What do you think? Is the option B correct? No, right?
Mm.
Right. So it's very difficult to get differentiated cells in large numbers. And also, even if you, by a very optimized method, you get it, these cells, they will not survive in culture, uh, like for different, like for a large number of cycles. In 3 to 4 cycles, they will start showing the signs of cell death. They will start undergoing senescence and then cell death. Their metabolism slows down. Okay, cell number stops increasing. So that is very common when you try to work with differentiated cells. That is why whatever experiment that you do with differentiated cells or whatever application that you have in mind, you have to think uh, like how many number of cells you need. Can the need of these number of cells be fulfilled with differentiated cells, or do I have to think about using stem cells? Because as I told you, if the stem cell is given proper environmental cue, it will its molecular pathway can be changed and we can trigger a cell differentiation pathway. Okay, and in the process, what happens? The cell undergoes changes in morphology, gene signature starts changing, and cell numbers also starts increasing. And ultimate product is that you get a large number of differentiated cells with identical functional and morphological properties. So, because of reasons stated in A, C, and D, stem cells are becoming very popular in tissue engineering. In tissue engineering, we need large number of cells, like it is there in our body. If you must know, our body is tissue that, like even a small section of it, actually contains 10 raised to the power 11 cells to 10 raised to the power 14 cells. Okay, our tissue or organ contains this many number of cells. So if you were to create any clinical benefit with your tissue engineered constructs, you need to at least reach this number of cells. Okay, like reach as close to possible, like as close to it as possible. Okay, and you cannot just rely on cell difference, like differentiated cells to divide and then they will reach. Usually we start with say 10 to the power 5 or 10 to the power 6 cells and differentiated cells. And by luck, even if you reach 10 to the power 11 cells, by then the activity of these cells will be very less. They will start dying. Okay. So that's why stem cells are very much preferred in this field. So this is the next question. Which of the following correctly highlight the challenge or challenges when working with stem cells? So now you, for your application, you have decided that you want to work with stem cells. But before you start working with it, you should know what problems can arise or what challenges you will face in the process. So first is, so what are the options here? S- Can it give rise to teratoma formation? What is teratoma? Teratoma is a tumor. Okay, teratoma is a tumor where you will find cells from all the three germ layers, endoderm, ectoderm, and mesoderm. Okay, so this is a challenge with embryonic stem cells or when you work with cells derived from fertilized egg. So these cells, if suppose some problem happens in their gene expression and something like that, or if the proper cultural conditions are not given, this teratoma formation is a possible um, effect that you can see, means it can happen. Then it's difficult to achieve differentiation in vivo. Yes, it's true for stem cells, by the way. So, uh, in vitro, people have successfully differentiated these cells, okay, into a specific differentiated cells by controlling the environmental factors very finely. But as soon as these stem cells in their native form are introduced in vivo as it is, it's difficult to mimic, it's difficult to control the environment in your body, right? Which is why after a few days, this characteristic of that cell changes into that of a differentiated cells. So it's difficult to achieve um, like or maintain its properties in vivo when you're working with stem cells. Then ethical issues. So obviously, ethically, there are so many questions when you start working with stem cells. Actually, you have to get special permissions from ethical groups who, ethical committees who are only sitting there to give you permission whether you can work with stem cells or not, because there are various ethical concerns. So these are very powerful cell types, and you can create various dangerous things as well, or unknowingly or knowingly. So there are committees there who are to judge whether you actually need these cells or not for the research. How much benefit will you bring to the society when you're planning to work with stem cells? So these are questions they ask you, these committees ask you before you get approval to work with stem cells. So these are the three challenges. All right, one or three like there are various others as well. These are just three of them when you are thinking to work with stem cells. So the correct option is all of the above. Okay. Um, Give me a second, okay? Let me share my screen again. This is the next question. Which scientist discovered that the retroviral expression of four genes, OCT4, SOX2, KLF4, and c-MYC converted somatic cells into a pluripotent state? So this work that is stated here is basically the first work in the field of induced pluripotent stem cells. And one of these people actually were the people who discovered this unique type of cell population. Can you recognize their names? Okay, no problem. So it is actually Takahashi and Yamanaka. So this is the paper that was published in 2006. It has over 30,000 citations. And in this paper, for the first time, these two people, Kazutoshi Takahashi and Shinya Yamanaka, so they are Japanese people, and they for the first time, showed that if you express these four genes, SOCT4, SOC2, KLF4, and c-MYC in a differentiated cells through a retrovirus. Okay, you can actually change the property of that differentiated cell into a stem cell. You can convert back. So you can actually, so usual mode of what happens in nature is that it is the stem cells who gets genetically modified in our body to become differentiated cells. But what they suggested in their paper is that you can take few differentiated cell population, okay? You express these genes OCT4, SOC2, KLF4, and c-MYC. It's they recognize these genes actually, and then they said that these four genes are the crucial genes which actually helped in reprogramming of the differentiated cells back to its pluripotent state or undifferentiated state or become stem cell. So this stem cell population is now what we call as induced pluripotent stem cells. Now people are working on this cell population to further differentiate it into other cell types like many cell types people are trying to multiple cell types. So, as I told you, it is very difficult to find these stem cell niches in adult tissues because they are present in very less number in compared to the rest of the cell population. Also, when we talk about embryonic stem cells or bone marrow stem cells, so there are various ethical concerns related to it. But it is easy to get permission to work with differentiated cells. So, this approach allows you to you know, face less problem in case of in terms of ethics. Ethically, this approach is safer, I would say. Also, many now methods are getting standardized to get pluripotent stem cells. So once you have these pluripotent stem cells, then you can take the benefit that this stem cell does have. Like you can get large number of cells, you can differentiate it along multiple lineages. So all those benefits of this working with stem cells you will get, as well as you will avoid the challenges that are there when you're working with, say, embryonic stem cells and all. So this is a very pioneering work by these two scientists. And if any one of you would come to research and all, nowadays induced pluripotent stem cells are widely used, right? So you may get opportunity to work with these kind of cells. Now, match the following. This is the match the following question. So given our options A, B, C, right? Depending on what you know about these type of cells, you have to correctly match. I'll give you a few seconds. Think a little bit. So anyone who would could think what matches well in this question? So what's the difference between multipotent, totipotent, and pluripotent? So according to lectures, multipotent means those stem cells who can undergo differentiation into multiple types of cell lineages, multiple, okay, two, three, four types, okay. So multiple, but note they cannot give rise to cell types that are found in placenta or embryonic cells, which are present during the early development stages. So those cell types cannot be formed, multiple cells. Totipotent means these cell types or these types of stem cells can give rise to cells, all the cells in our body, and including those cells which are there uh, during development, early stages of development. Okay, all cell types, total. Okay. Now, pluripotent means it again it cannot give rise to the early stages or cell types found in the early stages of development, but it is it is having more potency in comparison to multipotent. So here also, if multipotent stem cells can give rise to, say, two, three lineages, but pluripotent stem cells can give rise to more lineages in comparison to multipotent stem cells. Okay, so very briefly, I tried to explain. Now, if you have gone through the lectures, try to match these 8 options which are there in your right hand side with the 3 types of cells. Okay, we'll go one by one. Multipotent stem cells, which of the eight seems correct? Is it having high stem cells? Can you derive it from the inner mast cells of the blastocyst, or is it available in the early cells of the fertilized egg? Is it low? Does it have low potency? Can you call mesenchymal stem cells as multipotent stem cells? How high are the ethical concerns are there when you are working with multipotent stem cells? Can you isolate multipotent stem cells from adult tissue? Anyone? As I said, multipotent stem cells, they are having low potency in comparison to pluripotent stem cells. Okay. If you rank these cell types in terms of potency, totipotent stem cells have the highest potency because they can differentiate along all lineages. And next, you can rank the pluripotent stem cells, and then comes the multipotent stem cells. So it has the lowest potency. So the number of lineages it can differentiate um, is less. Okay. Then examples of these type of cells. Let me come to that. Is iPSCs or induced pluripotent stem cell? Is it a um, pluripotent stem cell or multipotent stem cells? iPSC, anyone? It is a pluripotent stem cell. Both iPSCs and embryonic stem cells are pluripotent stem cells. Okay? But they cannot give rise to the placental cells or early cells of the embryonic development. They cannot give rise to that. Okay. Then, which of the stem cells are derived from the inner mast cells of the blastocyst? Anyone? Embryonic cells, right? Embryonic stem cells are derived from the inner mast cells. So C matches with 3. Now, fourth, early cells of the fertilized egg. If you use those cells, what characteristic will it display? Totipotent, pluripotent, or multipotent? Well, actually, you can label them as or categorize them under totipotent stem cells, like the zygote. Okay, or a zygote starts dividing into 2, 4, 8. So these cells, if you take individually, they are totipotent in nature. Then mesenchymal stem cells. Let's say you isolate it from a bone marrow and all. So is it multipotent, totipotent, or pluripotent? Anyone? It is multipotent. Okay. Bone marrow, it has been shown that it can undergo differentiation and give rise to chondrocytes, osteocytes, adipocytes. Like these are the three main lineages that these multipotent stem cells can give rise to. Which of these cell types have very high ethical concerns related to their use. So with greater power comes great responsibilities, right? So which of these cells have the greatest power, you think? Totipotent, right? Totipotent stem cells and pluripotent stem cells. So these two cells will be having greater ethical concerns, correct? Am I correct or not? Are you following? Last, we are left with adult tissue. So this is the source for which of these type of stem cell you can actually get multipotent stem cells from adult tissues like your epithelial tissue, umbilical cord. So from these kind of tissues, people have isolated stem cells which can undergo differentiation along one or two lineages. That's it. Okay. So moving on, if you have any queries, feel free to ask and try to answer the questions. These questions are for you guys. If you have queries, ask me. Okay. Okay, this is the next question. What is the primary purpose of somatic cell nuclear transfer in the context of biological research? So this is the technique um, very commonly used to get embryonic stem cells. So here what people do is they take an egg, unfertilized egg or oocyte, remove its nucleus. This nucleus is removed. Okay, so this is now a cell without a nucleus. And they take a somatic cell or differentiated cell of, say, any specific tissue type. Now they take this nucleus and add it to oocyte. So this is nuclear transfer is happening from a somatic cell. So what it does, it basically genetically reprograms the further development of this oocyte. This oocyte, when it will, so the somatic cell is having all the 46 chromosomes. So now it is basically become a fertilized cell in terms of the chromosome number. So the further development of this oocyte will now give rise to different stages of cells like blastocyst will form, okay, where you will see inner mast cells, and from this inner mast cells you can isolate the embryonic stem cells. And if you allow further development, say you transplant it inside a womb of an animal or something like that, you can also successfully do cloning. Okay? You can create a person or an animal who's genetically identical to this somatic cell. So this is the technique, and this is what the question is asking. What do you understand about somatic cell nuclear transfer? So this technique is obviously a very powerful technique, and many ethical uh, concerns are related to here. So it is not very commonly practiced. So as I explained, depending on that understanding, please try to answer which of the option is correct. Option A, to combine genetic material of two different species in a single organism. To clone an organism by transferring the nucleus of a somatic cell into an enucleated X cell. To introduce foreign DNA into an organism. To create genetically modified organisms, GMO. To produce somatic cells with increased resistance to diseases. So, which of these option correctly defines the technique somatic cell nuclear transfer? It's option B, right? We are basically transferring the nucleus of a somatic cell of interest into a enucleated, means the nucleus has been removed of an oocyte or X cell. And when this oocyte is further allowed to develop, you can go up to like creating an entire organism. Okay, so this is the technique. This is the next question about asymmetric cell division in stem cells. So why this is important? That is what the question is asking. What is the significance of asymmetric cell division in stem cells? First, it ensures that both daughter cells become specialized and lose their stem cell properties. It results in the production of two identical daughter stem cells, each with the same potential to differentiate. It allows for the production of one daughter cell that remains a stem cell and another that differentiates into a specialized cell. It causes both daughter cells to become fully differentiated, leading to the depletion of the stem cell population. So which of these options fits the definition of asymmetric cell division? So those who were there from the beginning, I expect you to try because I've already explained what do you understand by asymmetric cell division in a previous question. Yes, if anyone is eager, please try.
Ma'am, is it option C?
Yes, it's option C. Good. So asymmetric means that one daughter cell will remain stem and other will undergo differentiation. That is how they maintain the population of stem cells. Okay, so it's option C. The next question, which of the following statements best describes the role of adult stem cells in the body? Adult stem cells means those stem cells that are derived from adult tissue. Okay, means yours and mine tissue if you use. And in that tissue, there is a very small population of cells with stem cell characteristics. When you use those stem cells, they will be now called adult stem cells. Similarly, you can derive it from a fetus, unborn fetus, or from an infant, newly born infant. So, depending on the age of the tissue, yeah. So the characteristic of the stem cell will definitely differ. Okay. So we are talking only about adult stem cells here. So which of these statements best describes its role in the body? They are capable of differentiating into any cell type in the body, similar to embryonic stem cells. They are found only in the bone marrow and responsible for producing blood cells. They maintain and repair the tissues in which they are found by differentiating into specialized cell types. They are primarily involved in the formation of the placenta during early embryonic development. Which of these options are correct? Anyone? What is the role of adult stem cells in the adult tissue? What do you think is the role? Okay, so let me make it easy for you. Option A is incorrect. Okay, so adult stem cells, they are multipotent. They are not pluripotent stem cells like embryonic stem cells. Also, these cells are not involved in the formation of the placenta. So either B or C is correct. Now tell me. Anyone?
Option C.
Option C. Okay, you are correct. It is option C. So adult stem cells, they are found in almost all tissues. Even they are found in your scalp. So there is a special cell type called hair follicle stem cells. Then people have also identified in urine. Okay, so these cells, these adult stem cells are present in almost all tissue type. Their primary role is to support the functioning of the tissue when in scenarios when it gets damaged, right? And there is a need of new cell population. So these stem cells, they are usually in an inactive state, and when such kind of situation arises, they wake up from their sleep and starts proliferating and uh, give rise to new differentiated cells. These cells will then go and help in repairing the damage in that particular tissue. So our body has a very nice regenerative potential in itself. Through tissue engineering, we are just trying to supplement it. We are trying to accelerate it even more so that quality of life can get improved. Okay, so option C is correct. So this is a bonus question. Okay, um, it is not compulsory for all of you to answer. I'll anyway tell you. But I would like you to try. Okay, I'll give you a few seconds. If you are able to try, then if you are able to answer it correctly, it's fine. Otherwise, I'll tell you the answer. Which of the following is a are an adult stem cell? Birth-derived hair follicle stem cells, adipose-derived stem cells, embryonic stem cells, induced pluripotent stem cells. Which of these is or belongs to the broad class of adult stem cell? Give it a guess, maybe.
Ma'am, can you please tell more about option A?
Okay. So, bulge-derived hair follicle stem cells are basically the cell population which are found at the tip of your hair follicle. Hair, okay? So, hair grows at different stages. Uh, and there is a phase called anagen phase, also known as the growing phase. When that phase comes, you can actually see this kind of bulge forming. And this is your uh, anchor into which the hair follicle is there. So in this phase, anagen phase, this bulge is very prominent, and it has been identified that few cells are present here, and these cells are basically helping. They migrate. They first migrate to down. Okay, they migrate down and provide more number of cells that helps in growing of this hair shaft. So these cell population are actually called the bulge-derived hair follicle stem cells. So as I told you, it's a bonus question. So actually, it's not compulsory for you to answer. The correct answer for this question is option A and B. Okay. So the bulge-derived hair follicle stem cells can be obtained from any person with hair, obviously. And if the person is having a very good hair, means very thick hair grows on that person. So it's the uh, and if you pluck a hair like with a tweezer or something like that, and from that end of the hair, you might see some white colored bulb. Okay, so if you collect more and more hair strands with this characteristic with white bulb, you can further process it and get large number of these stem cells. But if you do it with a person with very less hair growth or very almost no hair growth, then the cell population will be definitely less. Adipose-derived stem cells refers to those stem cells derived from adipose tissue or fat tissue. Okay, whereas the other two cell types are not from adult stem cells. As the name suggests, embryonic stem cells are derived from the inner mast cell of the blastocyst. In this pluripotent stem cell, as you know, is reprogrammed. You are reprogramming a differentiated cell into a stem cell property that is in iPSC. Okay, moving on. Which of the biophysical material properties can direct cell differentiation? So there are some biological properties and there are some material properties. So material properties, why it is important for us to know? Because we are ultimately going to seed these cells or grow these cells on some biomaterial. So those material properties can also affect whether the stem cells can undergo differentiation on that substrate or not. So this question is actually quite easy. Option A states activating endogenous transcription factors. Is this a material property? Yes or no? Okay. Co-culturing with specific cell lineage type. Again, the same question. Is it something to do with the material that you have used? Substrate stiffness and then substrate topography. Are these two options are one of the material properties that you have looked into before? Or and which of these options is correct for this question? This question states which material property or biophysical material property can actually influence the cell differentiation. So option A and B definitely is not something that a material related to material property, right? Because in option A is saying that you are using some retrovirus in order to activate the transcription or some transcription factors, okay? Or you are using some biological molecule to trigger the activity of some endogenous transcription factors. Option B is saying that stem cells are grown in presence of another type of cell. So through cell-cell signaling, the cell differentiation can be directed. So these are some biological properties, or you can say manipulation or control biological properties you are controlling in vitro to direct cell differentiation. Whereas substrate stiffness and topography depends completely on the material. What's the Young's modulus, whether the substrate is rough or smooth. So depending on that, the cell behavior will change definitely, as we all know. That can also have effect whether the cells, it will definitely affect which lineage, under which lineage the cell differentiation is undergoing, right? That will also affect it. People have reported this kind of work. So option C and D is correct here. Now, this question is on the technique flow cytometry. What is the primary function of flow cytometry in cell analysis? Flow cytometry, what is it? Okay, it is what it is asking. Means what do you understand by flow cytometry? So option A is to separate different types of proteins based on their size and charge. Option B, to measure the physical and chemical characteristics of cells or particles as they flow in a fluid stream. To sequence DNA by analyzing the nucleotide order in a genome, cell's genome. To amplify small segments of DNA for further study. So after reading the options, what do you see? Option A basically refers to the technique of Western blotting, don't you think? Means you are working with proteins and you want to differentiate it, differentiate the protein types based on its size or charge, or some similar to some kind of protein electrophoresis. SDS, right? SDS page is used for proteins. Like for DNA, gel agarose electrophoresis is used. For proteins, SDS page is used. Their detergent is used SDS, and on a polyacrylamide gel, electrophoresis is done. So option A is referring to SDS page. Now, option C, what it is saying, to sequence DNA by analyzing the nucleotide order in cell's genome. So this option seems like it is referring to Sanger sequencing, right? Everyone knows what is Sanger sequencing. So it's a technique through which you can understand or determine the sequence of your DNA sequence, like what is the ATGC, what is sequence of these nucleotides on a DNA strand. You can determine that. Whereas option D, as the statement says, is just PCR. Okay, you're amplifying a small segment of DNA. So neither option A, C, or D is correct. So this leaves option B. So what option B is saying, to measure the physical and chemical characteristics of cells or particles as they flow in a fluid stream. So this is what is flow cytometry. So what this statement say, let me help you understand. Flow cytometry can be done for particles as well, small particles which are of size similar to cells. First point. Second point, here cells in a suspension form is loaded into a machine and flowed such a way that a detector can actually detect or analyze one cell at a time. And as it is doing so, it will give you some data related to that cell type. That is what the statement is saying. So again, I'm repeating. You are using a cell suspension and it is flowed through a tube, very narrow tube. Here, one cell at a time, they are flowing, and there is some detector, okay, which can analyze one cell at a time, detector, and it will give you some data, some graph or something like that, some data about the cell type. So in a software, you will see that you will see a cumulative data of about all the cell types. Okay, that is flow cytometry. Now, how does this happen? How the detector is recognizing the property of the cells? Okay, so people use antibodies. Okay, and antibodies are bound to specific surface markers or antigens on the cells. These antibodies are themselves be tagged with some fluorescent dye. So there might be a light source here, say light source, and it is maybe giving light of two wavelength, wavelength 1 and wavelength 2. So you have two populations of cells, suppose, and one population of cell, suppose, express antigen 1, another population express antigen 2, and you have specific antibodies for antigen 1 and antigen 2. And on the tail of antibodies, there is a fluorescent or fluorophore. And this fluorophore will give fluorescent light in, say, wavelength, in presence of light wavelength 1, this is in presence of light wavelength 2. Now you are specifically, what do you say, tagging the two cell populations with antibodies conjugated with a fluorescent dye. Now, as these cells, so in the suspension, you cannot say, right? All this populations of cell one go first, then cell two population will go. You do not have control over it. You will have a heterogeneous mixture of cell. Now you want to know how much percentage of population one cell type is there and how much percentage of population two cell type is. That is what you want to know in your study. So you pass that heterogeneous cell population through this flow cytometer machine. There, the depending on the what type of fluorophore is there, some fluorescence will come. That fluorescent signature fluorescent will be detected by detector, and you will then get a data. And that data you will see graphs, and from that graphs you can estimate the percentages. So this is how very general a flow cytometer works or flow cytometry does. So this is just analyzing the cell population here. You can also do sorting, cell sorting, by the way. Okay, cell sorting is also possible. So flow cytometer with FACS or cell sorter. So FACS is fluorescence acid, fluorescence activated cell sorter. My pen's battery is down. Yeah, that is why I'm not able to write. So FACS is fluorescence activated cell sorter. So flow cytometer with FACS also come. It's a special attachment with the machine, and they cost a bit more. But through this machine, you can actually get fractions of different cell populations, and that you can use further for cell culture. Okay, so it's a method of purifying your cell heterogeneous cell population even further. You will get very high purity of cell population that you can use for tissue engineering application. Okay. So I hope now you're able to understand what is flow cytometry and what is FACS or FACS with flow cytometry. Okay. Now, which of these cell isolation technique is not based on surface antigen binding principle? So FACS, as I told you, um, you are using antigen here, right? In the previous slide, I told you FACS with flow cytometry are using an antigen to and specific to that antigen, antibody is used. So antigen antibody binding principle is used in FACS. What is MAX now? Anyone? So F stands for fluorescence, MAX stands for M here. And MAX in M stands for magnetic. Okay. So MAX is magnetic activated cell sorter. So here the antibodies are actually tagged with some magnetic beads. And here you are not using a light source, rather some magnetic field in order to um, direct the cell population, which is tagged with the uh, magnetic bead of a particular polarity. Depending on the polarity of the magnetic field bead, your and your field that is there, magnetic field you there, you can sort the cell population. That is max. Here also, you are going to use density. Here also, you're going to use the surface antigen binding principle. What is aptamer binding? So the correct answer is actually showing, it's no problem. So here density gradient centrifugation is the correct answer. I'll come to it later why it is the correct answer. What is aptamer binding? Aptamer basically refers to small single-stranded DNA or RNA molecules, which, like antibodies, can specifically bind to proteins, carbohydrates, etc. Those are called aptamers. So aptamers you can design, okay, and make it specific to the specific and surface marker on the cell. So it is not protein origin. Only difference is a polynucleotide, single-stranded RNA or DNA, which can fold into specific configuration as it is single-stranded and has a very specific binding affinity towards a protein or carbohydrate. That is aptamer binding. Whereas density gradient centrifugation, here what happens, you are isolating cells based on its density. You use a medium, okay, where different densities. You can make different densities. And when you do centrifugation, cell with uh, matching densities of that particular medium will be found. So you create a column, suppose, or a tube with different kind of materials, different kind of liquids of different densities. And since they are having different densities, they will not mix. So you have created a column like this. And top of it, you add a cell suspension. So depending on which density medium the matches the cell's density, after you centrifuge that entire system, you will see the cells will be present in that density level. Okay. And when you isolate the density level, you can further purify it. So it will help you, this technique will help you to isolate cells based on density. But please note the purity of the cell population is not that high. Means you will have some cell debris or few cells of other population present in your purified product. You have to go to higher level of purification method in order to get only homogeneous cell population. Okay. So this is the next question. Which type of cells usually are obtained from bone marrow? So, from bone marrow means you are talking about the adult stem cells. So, which of these are adult stem cells? And adult stem cells are multipotent in nature. We have already seen this question, like a variation of this question previously. So, answer from you guys would be nice. Anyone? Those who have been in this session from throughout, from the beginning, actually. Why don't you give it a try? It's a simple question. We have already discussed a few variants of this question previously. So, mesenchymal stem cells are obtained. Okay, from bone marrow, you will get mesenchymal stem cells. This is also called bone marrow mesenchymal stem cells. From bone marrow, you won't be able to get adipose-derived stem cells or embryonic stem cells or pluripotent stem cells. And these mesenchymal stem cells are multipotent in nature. We have already seen this previously, right? Please try, okay, and pay attention. You'll be able to answer, not an issue. Now, which of the following cell isolation technique is based on cell size? So you are isolating cells of your interest based on its size. Which of this technique will be useful in doing so? Aptamer binding, as I told you, is based on surface antigen binding. Max is also based on surface antigen binding. But what about plastic adhesion and filtration? Anyone? Plastic adhesion means you're basically using a plastic substrate to adhere to make your cells adhere. Yes, even yes, anyone.
Ma'am, is it option B?
Yes, it's option B. Okay. So plastic adhesion will not allow you to isolate cells based on size. Rather, it will help you to isolate cell based on its ability to adhere or not. You can isolate suspended cells from the adherent cell types. Okay. The filtration, where you will use some porous membrane of specific size, of specific pore size. Okay. Usually, cell strainers are available for this work. Very small, like a funnel, like a tea strainer, the thing looks like very small, and it can fit into your well plate, having membrane of some particular pore size. And through using these cell strainers, you can filter out your heterogeneous cell population mixture. Okay, and you have to choose your pore size depending on the cell type you're targeting. Okay, it should be definitely it should definitely just allow the passage of your cell type of interest, whereas cells which are larger than this pore size will stay on the film, and cell of your interest should have size less than this pore size, then only it can pass through. That is the principle, basic principle of filtration. So option B is correct. Which of the following cells are adherent in nature? Anyone? Anyone?
Ma'am, I think
Adherent.
it is option D, RBCs.
RBCs are not adherent. RBCs, they remain in suspended form in our blood. Same with our immune cells, like natural killer cells and T lymphocytes. It's option C. It's the fibroblast cells that
Okay,
can
okay,
adhere on a substrate. Okay? Yes.
okay.
Now, next question. Which of the following is a method for cell harvesting? Cell harvesting means you have to get cells from your tissue, right? From your organ. So you have to first get a piece of your tissue. And from that piece of tissue, you will isolate the cells from the ECM. That's it. Here, when your goal is to remove the cells intact, okay, without killing it, and the ECM should be removed. But when we talked about decellularization, there our goal was to get the ECM in intact form. Doesn't matter if the cells are getting damaged. Okay. Here, our goal is to get cells in intact form even if we have to like degrade the ECM. So that's the objective here. But the starting material is a tissue. So which of the following methods, A, B, or C, or D, will allow you to get the tissue sample from your body? Punch biopsy, bone marrow, endoscopic biopsy, all of the above.
Option D, right? Because we are
Yes,
getting cells
correct.
from all of these.
Yes, yes, it is option D. So biopsy means you cut out a small section. And all of these techniques that I've written in ABC, these are all clinically used, okay, for and you and I cannot do it. You need specialized surgeons who are expert in surgery at different parts of your body. They will help you get that small biopsy sample, and they are not routinely done on any patient. Uh, like we have to ask the doctors if they have a patient who is going to undergo surgery of this section or organ, and if they agree to help us out, then we have to get some consent first from the patient, and then the surgeon will while surgeon is doing his job, like maybe some repair work is needed through surgery. During that work, that person, that surgeon, will give us a small tissue sample. Now, as a researcher, what I'll do, I'll use this tissue sample to get the cell of interest and use it in my work. So that is how you will get a biopsy. You and I, we are PhDs or we work in research institutes. Okay, we are not qualified to get these biopsy samples. It is the doctors and the surgeons who have to help us. So this field of tissue engineering is very interdisciplinary, as I told you in the very beginning. People from various expertise are required in order to advance this field. Okay. So that is why there is so much to learn.
Ma'am, I have a
Now,
follow-up question about that.
yes, yeah.
So can it be like an isolated procedure, or does it always have to be a part of another procedure, like getting the biopsy? Can it be just an isolated procedure to just obtain the cells?
So usually you cannot convince someone to undergo surgery for research. Okay, it's very difficult uh, to convince someone. Usually, it's easier, if the doctor already has a surgery scheduled for a particular patient and the patient needs that surgery. In those scenarios, it is usually easier. You cannot convince a person to undergo surgery. Just you can get a small tissue sample. It's very difficult.
Okay, got it. Thank you, ma'am.
Yeah. Which of the following, next question is the next question I'm reading. Which of the following methods is used to mobilize stem cells from the bone marrow into the bloodstream for the collection of peripheral blood stem cell harvesting? Okay. So usual route of bone marrow or usual procedure of isolating bone marrow is very painful. Okay. They drill into your tibia, that is the femur bone, that thigh bone. The top part is which is connected to your hip and socket joint. There, the bone marrow cells are found. Also, in the pelvic region, the iliac bone is there. So, iliac bone also contains bone marrow. So, these are uh, like very hard tissues. So, in healthy person who is not undergoing total hip joint replacement, means his hip joint is healthy. If you want to isolate from that healthy person the bone marrow, then the doctor has to drill into the uh, skin and then into the bone using very drilling tools and all. And then it reaches a very soft portion. So in that soft portion, they insert a needle and aspirate it out. And doctor has to be very careful not to aspirate out too much of bone marrow because bone marrow is also present in very small volume. So less than 2 ml of bone marrow aspirate is taken out. So this procedure, as it sounds, is very painful, and the person has to be given either local anesthesia or complete anesthesia. Okay, but there is another less painful, less invasive procedure to get bone marrow. That is, you can inject a growth factor or some chemical into the blood. So that name I'll not tell you the answer, otherwise I'll tell you the answer right away. So you have to first inject that chemical into the bloodstream, and that chemical will signal the bone marrow stem cells to come out from its niche. Okay, it will come out from bone marrow. Okay, and start uh, floating in your bloodstream. And then you, just like you draw out blood, you can draw out the blood with bone marrow stem cells. So but this process again will not give you a very pure bone marrow aspirate as it sounds. You will get a mixture of other cells like the blood cells mixed with it. You have to use specific isolation procedures, purification procedures, and then FACS or MAX in order to get very high yield. But the yield, in order to get very pure yield, but the less number of cells you will get if you do this peripheral blood stem cell harvesting, right? The yield will be very less, um, and the purity of the cells will be also compromised, right? In comparison to the usual or the conventional method. So, which of these chemicals that is listed in A, B, C is used? That's the question here for this particular way of isolating bone marrow stem cells. Option A is chemotherapy. Option B is growth factor injections, such as granulocyte colony stimulating factor. Okay, or it is also called uh, granulocyte macrophage colony stimulating factor, GMCSF is also called. Then radiotherapy, immune suppression therapy. Which of these options seems correct?
Um, I This is just a guess, but I think option B.
Yes, you guessed correctly. It's this option B. Doctors inject this GMCSF into the blood. So it is a cytokine, by the way. So very well-studied cytokine and is involved in triggering cell migration. So what it will do? It will trigger the bone marrow stem cells to migrate out from bone marrow and come into blood so that the doctor can easily aspirate out the blood containing bone marrow stem cells. Okay, so it is option B. This is the next question. Which of the following techniques can be used to retrieve cells from skin tissue biopsy? Okay, so skin tissue, skin, and you say use a scalpel to cut out a small section of skin, or you use a puncher, the puncher that we use to make holes in our body. Similarly, punch type of thing are available, instruments that can create a small circular cut on your skin and remove it out, and will create a very small wound that will heal in few days. That is punch biopsy. So suppose you use that machine, that instrument to create a small skin tissue punch biopsy, and then from that skin tissue you want to remove the cells. Okay, you want to degrade the ECM in some way and retrieve only the skin cells. So which of these options seems a good way to do that? Acid digestion, scraping cells off with a scalpel, enzyme digestion, all of the above. So skin tissue contains, let's say, so skin is a multi-layered tissue, and in your biopsy, let's say you have only removed the epithelial layer. So topmost is the epithelial layer, the layer that you can see right now on your body. The bottommost layer is the dermis layer. Okay, as you and the biopsy can be done in such a way that all the two layers are removed or only one layer is removed, depending on how deep you cut. So suppose you remove the epithelial layer. The epithelial layer contains the keratinocytes and some proteins like keratin, okay, that are holding the cells together. So which of these option now seems good to remove the keratinocytes from the protein-based ECM? Yes,
Is it B?
is it B? Okay, so if you use B, then what will happen is cells are present in very high number. Okay, and you won't be able to get pure cells. So it's not a very hard surface as well. The tissue that you will get is not very harsh. Hard. It's not very hard surface. So if you try to scrape the cells out, then what will happen? You will only get small population of it. And like you may even end up rupturing it, the biopsy. You may even end up rupturing the biopsy. So it's not a very efficient technique. Okay, although it can work. Okay, I'm not saying it won't work, but it's not very efficient if your goal is to get large number of cells. If you see at option A, acid dissection, the technique uses acid and it's a very harsh, like maybe some HCl kind of acid can be used. So it sounds good when your tissue is having some mineral in it, like bone tissue. So it can corrode that mineral component and get the cells. So that kind of property is not present in skin tissue. So option A and B is not good. Whereas if you use enzymes like keratinase, collagenase, okay, so these kind of enzymes will act on the proteins that are there in your skin, like collagen, keratin, etc. Then what will happen? This ECM component will degrade in a much more gentle manner. And these enzymes, they will not harm your cells that are residing. So you will get a suspension, okay, with enzymes. When you add the enzymes into your tissue biopsy, incubate it for a period of time, what you will see, the ECM will start coagulating towards the bottom, and maybe the supernatant, if you centrifugate, you will get a supernatant. So supernatant will have the cell population. So it's much more gentler, okay, and you will get a higher yield. So enzyme digestion is the correct answer here as well. Okay. Yes. Doubt? A doubt is there?
No, no. I just wanted to confirm once again that you said if we use acid that is generally used for hard tissues, right?
Yes, yes, yeah.
Okay, okay. Yes, ma'am. Thank you.
This is the next question. A research scholar wants to select adipose stem cells. Okay. So he or she used an antibody that can bind to the surface marker of the stem cell to sort and isolate it from the crude cell population. Choose the right option that fits the above statement. So the technique that the research scholar has opted, uh, will it come under positive selection method or negative selection method? So briefly, what is positive selection? What is negative selection? Positive selection means you are interested. So in both cases, positive, negative selection, there is a heterogeneous cell population is there. You have a mixture of cells. Okay. In positive selection, you want to remove or isolate your uh, interest, your cell interest. Okay, means yes, the cell that you're interested in, you only want to select that out of say 10 cell types. That is positive selection. Whereas negative selection in a mixture of cell, what you do apart from your cell of interest, you kill, you somehow cause the remaining cells to die, okay, by giving some condition. And in that way, you isolate your cell of interest. So let me repeat. In positive selection, you're isolating your interest cell type of interest by not harming the remaining cells. The remaining cells are as it is. Okay, you're just picking out from a if there's a bag containing red blue balls, and you're just picking out the red balls. That is positive selection. Whereas a negative selection in a bag, in order to get the red ball, you what you did, you say burst out all the remaining balls, and that leaves you with what? The red ball. So that is negative selection. Okay. So the research scholar, what he is doing, he wants to select only the adipose stem cells. Okay. And suppose he has a mixture of cells and he wants to select out of this mixture the adipose stem cell. So for this, he is using antigen antibody binding. The antigen here is the marker specific to this adipose stem cell. And to this surface marker, he has used an antibody. And this antigen antibody binding is very specific. So it will target only the adipose stem cells. Non-specific binding will not happen. The remaining cell types will remain as it is. Okay, they will not get sorted out or isolated. Only the adipose stem cells will get sorted out due to antigen antibody binding. So the person may be using the technique PACS, fluorescence activated cell sorter or magnetic activated cell sorter to isolate this adipose stem cell from crude cell population. So now this approach, under which category will you place it? Is it a positive selection or a negative selection happening?
This is positive selection.
Yes, that's correct. It is positive selection. Okay. There's another question here. You have a small cartilage tissue biopsy sample. Okay. The tissue that you have is a cartilage tissue. And you're interested in isolating chondrocytes from this sample. So which of the following seems like a logical way to proceed? So these are the steps given to you. Option A is enzymatic digestion, cell filtration, alcine blue staining, FAS. So what is alcine blue staining? Alcine blue is a type of stain that specifically stains the chondrocytes blue in color. Okay, so it is it can help you identify the chondrocytes very specifically. Okay, because the chondrocytes will stain blue. You can see it under light microscope. Then option D is cell filtration, enzymatic digestion, FACS, alcine blue staining. Enzymatic digestion, cell filtration, FACS, alcine blue staining, and option D is FACS, alcine blue staining, enzymatic digestion, and cell filtration. So which option gives you the most logical series of steps to get the cell of interest, that is chondrocyte? So you are starting with the chondrocyte tissue biopsy sample. Okay.
I am actually confused. I think it could either be option C or D.
Okay. So actually the correct answer is A. Okay. I'll tell you why. So first you have a chondrocyte, you have a cartilage tissue. So cartilage tissue mainly consists of collagen as the ECM and very small population of chondrocyte present in it. So cartilage has collagen. So you can use collagenase enzyme first to degrade out the collagen. All right. Then what you'll be left with? You'll be left with cells and some cell debris and all. Okay. Some erased cell if possible, like, or some contamination cells, like cell of different origin will also come in. So in order to only remove your chondrocytes from the rest of the components, unwanted components, you will then use a cell screener having a membrane of specific spore size. So through that, you filter out your mixture and you will get your chondrocytes. But again, to identify whether the chondrocytes are there or not, you can use alcyn blue staining. Okay. So alcyn blue staining and FACS, they can be like forks. Like after filtration, you can either directly go to FACS in order to, you know, specifically or increase the purity of your sample, as well as parallelly you can test the uh, presence, like in your sample, the chondrocytes are present or not. Like it can happen parallelly as well. These two steps can happen parallelly. It does not have to occur one after the other. Okay. So even if you use option C, in that case, it will be correct. Like you can filtrate first and then do FACS. Okay, that is also correct in that way. But D is not correct. Okay. A or C, either is correct.
Okay, got it.
This is another question. Which of the following surfaces promote cell adhesion? So here, cell adhesion is possible in which type of surface? That is what the question asks. Can cells adhere on a glass surface? Then what about polystyrene surface? Next is oxygen plasma treated polystyrene surface or none of the above. So cell adhesion with the onto a substrate heavily depends on the wettability of that surface. So if the surface is hydrophobic, very hydrophobic, okay, this if the surface is very hydrophobic, it will not allow cell adhesion. And if the surface is very hydrophilic in nature, then also cell adhesion will not happen. The key to achieve cell adhesion is to balance these two, hydrophobicity and hydrophobicity. These two properties has to be balanced. Okay, then only cell adhesion will happen. So glass surface, uh, is it hydrophilic or hydrophobic in nature?
Glass is hydrophobic.
No. If glass is hydrophilic, it's hydrophilic in nature. Okay. If you
Okay.
so the lenses that we wear usually, they are coated with some hydrophobic coating so that water doesn't stick. But if you use a naked glass, like naked, like without any coating, it is highly hydrophilic in nature. Okay. So if
Okay,
you
okay.
cast a drop of water on the glass surface, it will become flat immediately. It will spread immediately. That is why it is hydrophilic. Whereas polystyrene surface, what do you think? Is it hydrophobic or hydrophilic?
Um, can you give me an example of a polystyrene surface? Because I can't directly like think of anything.
So polystyrene is actually a uh, aliphatic polymer. Okay, quickly, it's like carbon and hydrogen only there. There is no uh, electronegative atoms. Okay, so generally polystyrene is not readily available in our society. Like we don't use polystyrene bottles. It's a type of plastic like PET, polyethylene tetrafilate, PET bottles, plastic bottles that we use to drink water. Like that, polystyrene is also like that, but it is having only carbon and hydrogen in its structure. If you quickly see the structure, Google it, you will see the structure of polystyrene. Based on the structure, can you guess? Will it be hydrophobic or hydrophilic in nature?
Hydrophobic.
Yes. So I just quickly Googled myself. Uh, Polystyrene is actually used to make tiffin containers like this. So let me show you my screen. Here. You must have seen this kind of tiffin containers. They are not readily available in India, but in abroad for packaging food, these kind of polystyrene based tiffins are used for taking remaining food from restaurants. And this is what the structure looks like. Okay, it is
Ma'am, your
completely
screen
having,
is not visible.
yes, yes,
Ma'am, your screen is not visible.
okay. Yeah. Is it visible now?
Yes, yes.
So yes, I was indicating to something like this. Polystyrene, this is the structure of the polystyrene, and this kind of tiffins are used. That is it. This kind of plates and tiffins are used. They are made up of polystyrene.
Okay,
Okay.
got it. Yes,
Yeah.
ma'am.
So they are actually hydrophobic in nature, right? So either of the surface cannot promote cell adhesion because they are towards extreme edges. But when we treat this polystyrene surface with oxygen plasma, then what happens? We introduce some hydrophilic moieties like oxygen onto the structure of the polystyrene, because of which some degree of hydrophilicity is also introduced. So that is why oxygen plasma treated polystyrene surface can promote cell adhesion. The cell culture flask that we use to culture adherent cells, they actually have this coating of polystyrene that is treated with oxygen plasma. And the speciality of using this polymer for cell culture is it is optically transparent and it's very useful. We need transparent surfaces for culturing cells because we often add some pH indicators whose colors change with change in pH. That helps us to monitor the pH of the culture media. If we don't do it, otherwise, very acidic or very basic pH will kill the cells, right? We have to maintain 7.4 pH. So for maintaining that pH, we indeed add a colored pH indicator and to visual and to clearly see the color and other things and optically transparent surface is needed. So if we use only plastic, naked plastic without this coating, also cell adhesion won't happen. Okay, we have to treat it with oxygen plasma, introduce some hydrophilicity, then only cell adhesion will happen. The main application of this coating is seen in cell culture flask. Okay. Now, this is the next question. I think it is the last question for today. I think so. No, there is one more question after this. Okay. This is second last question for today. This is again based on the understanding of positive and negative selection. So for the isolation of suspended cells, ultra low attachment plates are used in absence of serum. As a result, the adherent cells died, but the desired cells survived as a single cell suspension. This method of cell isolation can be classified under positive or negative selection. What do you think is the correct answer?
This is negative because the cells are
Yes,
dying.
yes. It is the negative selection. Okay, so we'll move forward as you have understood. So this is the next question. Facts of fluorescence activated cell sorting uses dash in order to isolate specific cells. Surface markers, density, cell size, or morphology.
I'm assuming that because it is fluorescence activated, it must be surface markers.
Yes. So here, surface markers are used to identify the cells. We use antibodies which specifically bind to these surface markers, and these antibodies are tagged with fluorescent dyes, okay, through which we are able to sort the cells. So that's it. Okay, that's it from my side for today. I hope the questions were useful to you. And if you have any queries, let me know. Like previously, if you missed any question or couldn't understand the logic of any particular question, let me know.
Ma'am, actually, I missed the starting few questions because of some internet issue at my end. So,
Okay.
like, uh, I think I will go through the recording once it is available, and then if I
Yes.
have questions, then I will ask you in the next session, ma'am.
Yes. Okay. That works. That works. Okay. The recording I'll upload it by tomorrow morning. You will find that in my YouTube channel. Okay. If any doubt
Okay.
you find, you let me know in the next session. Okay.
Yes, ma'am.
All
Sure.
right.
Thank you.
Thank you. Thank you for joining. And I'll end today's session. I'll meet you next week.
Yes, ma'am.